We are going to visit 3 classrooms in 3 different schools. We start our journey today at Alexis I. duPont Middle School in the 6th grade classroom of Danielle LaRosa. In 6th grade, students learn about Earth's place in the universe and the solar system through a space unit. Come on, let's take a look.
I really like how you guys came in nice and quietly. You're starting your do now. So I'd like for you to write down 2 to 4 sentences of what this quote means to you. America has tossed its cap over the wall of space. So I'd like for you to write down what that means to you.
So, Ms. LaRosa, what are we going to be seeing in today's lesson? What's today's lesson about?
Today's lesson will be about exploring the phases of the moon, as well as what's going on the moon currently with retroreflectors and just all the fantastic advancements that have been made through science and technology. And what you'll do is you're going to go over when I tell you to. We're going to walk very close, very slowly and orderly over there. You will get a styrofoam ball and you will get a skewer, all right? And you're just gonna carefully put this together like that. So right now, very slowly, we're gonna turn just a quarter turn so we face east, okay? Everybody go a quarter turn and look at the light on the styrofoam moon as you turn. Now, we're gonna turn some more. You notice the light gets larger. It's going from a waxing crescent into what's called a gibbous moon. We're going to keep turning. We're going to stop where it says north over there. This time, the moon is full. Can you see how it's completely lit up right now from where you're standing? All right, that is a full moon.
Ms. LaRosa, can you tell me what is it that engages your students to explore science?
I think using hands-on activities, lots of labs, and lots of technology really helps my students to explore science. So what you're going to do here is, these are different scenarios here, and I'm going to go through each one with you so you get an idea about what you can do, all the different options you have. The Red Clay Consolidated School District provides the best activities and technology for our students to really do well in science and understand all the complex topics. Good job. Give yourselves a round of applause.
Now let's explore the different phases of the moon even further with a quick lesson from our friends at SciShow Kids.
If you ever look up at the moon in the night sky, say before you go to bed, you've probably noticed that as the days and weeks go by, the moon looks like it's changing shape. Some nights, it's a little sliver, and some nights, a few weeks later, it's a big bright circle. Well, the moon doesn't actually change shape, but our view of it does. Why? Well, the moon, just like the Earth, has a day side and a night side, a part that's facing the sun and a part that's not. So when we see a little sliver of bright moon, we're seeing a little part of the moon that's facing the sun, while the rest of the side that's facing us is dark. And the size and the shape of that sliver of lit up moon changes because the moon orbits or moves around Earth. It takes the moon about 1 month to make 1 full orbit around the Earth. And as it moves around our planet, the daytime side of the moon, the side that's lit up from the sun, is facing in different directions as we see it from Earth. This means that every night we get a slightly different view of the moon. And during its month-long trip in orbit around the Earth, the bright part of the moon appears to grow and then shrink from right to left. The different shapes that we see of the lit up part of the moon are called phases. And there are 8 phases of the moon. The first phase is the new moon. This is when the nighttime side of the moon, the side that's facing away from the sun, is what we can see from Earth. The moon is still up there, but because it's the dark side that's facing us, we can't see it during the new moon phase. The next phase is called the crescent phase. This is when the moon looks like a little sliver, kind of like the tip of your fingernail. After the crescent comes the quarter moon. This is when the moon looks like a half circle. And the next phase is called gibbous. gibbous. A gibbous moon is when it's missing just a little sliver of light and it's almost full. And that's what's next. The moon is at its brightest at the full moon. That's when the fully lit up side of the moon is facing us, so we see a big bright circle. But we're only halfway through our phases. After the full moon, the lit up part of the moon appears to shrink. And just like how it grew from right to left, the sunny side of the moon will shrink from right to left too. So after the moon is full, it starts to shrink back down to a gibbous moon. But this time, you'll notice that the left side is shining bright instead of the right. After the gibbous phase, the moon will look like half a circle when it reaches the quarter moon phase again. And then after a few days, it shrinks down to a little sliver again, the second crescent moon. After that, the tiny sliver shrinks all the way down until the lit up side of the moon can't be seen at all. Now the moon is a new moon again. It's the same old moon, but it's gone through a month-long cycle of phases, and it's ready to start all over. Like I said, it takes a month for the moon to go through all 8 phases. In fact, our word month comes from the word moon because it takes a month for the moon to go from new moon through all of its phases and back to the new moon again. So tonight, before you go to bed, take a look out the window. If the sky is clear, can you see the moon? If you can, what phase is it in? Is it the bright part of the right side or the left side? As the days go by, keep an eye on it and watch how the moon looks like it's changing shape. You can even make drawings to keep track of how it changes from day to day. Now you know that the moon doesn't really change shape. We just see different amounts of the sunny side of the moon as it makes its monthly trip around the Earth. Thanks for joining us today. We love getting questions here at the fort. So if you have one, ask a grown-up to help you. Leave a comment down below or send us an email to kids@scishow.com. Thanks, and we'll see you next time.
We'll take some volunteer readers today. Danica, please start us off with our first paragraph. So today, the students that came in, and we did a do now that was a review of what we've been talking about the past couple of weeks. All right, once you write this heading, you want to cut out, what is this that I'm pointing to? Cell, animal cell or plant cell? Animal cell. Raise your hand if you can tell me how do you know that this is the animal cell and not the plant cell. Go ahead, Elijah. By the shape. They just completed their lesson 7, which is about cells in the TCI program. And so we wanted to reflect on that. We started out there today, and then we followed up with class notes, which we typically do. A lot of them really enjoy writing out class notes. And so then we want to add some images into the class notes so they can see what they're learning about. The next thing we want to do is cut out the second picture and glue that into your notes. Next thing we want to do is cut out the second picture, okay, and glue that into our notes. And we'll talk about what's in this image as well. Give you guys about 3 minutes to glue that in. Yep. And then vocabulary words. So it's always good for them to know the different terms that comes with what we're talking about. So we add that in today as well.
Learning about genetics is tricky because we rarely actually see the objects discussed. But remember, DNA, genes, and chromosomes are real physical materials, and each of these materials is located in every cell of your body.
Whole class, how many pairs of chromosomes do we have in humans? 23. How many total chromosomes do we have? 46. 46. Nice work. So they'll get some clay. All right. And then they need to make sure it should be about 8 centimeters in length. They're very interested in learning science through things that require them to work with their hands. It's great to see how TCI incorporates hands-on activities. Today we're working on an activity that requires them to create chromosomes representing of the human body. So it's hands-on experiences, keeps them engaged.
We should get something to cut this.
Does anyone have number 7? Okay. end off with an exit slip just to tie everything together. So I can get a general understanding of how much they know, and then I can figure out which students I may need to work with more on the following school day, which students totally understand what we were talking about.
As Miss Finney's class continues to explore DNA, let's ask a biologist about DNA, RNA, and gene editing.
Gene editing. You may have heard it linked to talk of GMOs, genetic enhancements, or even designer babies. But what is gene editing? And how does gene editing work to let us modify plants, animals, or possibly people? To answer these questions, let's take a closer look at DNA, RNA, and one of the best tools for editing genes, CRISPR. So what is DNA? Well, all of your genes are made of DNA. It is the instruction manual that tells your body how to develop and how to work. All of the DNA in a cell is called the cell's genome. A copy of the genome is found in most of your cells in a part called the nucleus. It's your DNA that controls many of your traits, like your eye color or how many toes you have. To control those traits and to keep your body working, the information in DNA needs to reach other parts of the cell. But DNA can't leave the nucleus. So how can those instructions be used? There's another molecule that helps, called RNA. RNA is similar to DNA, but it can send those DNA instructions out of the nucleus. Working as a sort of copy of the instructions, it can move to other parts of the cell. This lets your cells use DNA instructions to create proteins that keep your body working. Now, as you grow, your cells divide. Each of these new cells needs a copy of your DNA instructions, but the copying system isn't perfect. Sometimes mistakes are introduced during copying. How do these mistakes happen? If we look at DNA at a microscopic level, it has 4 different types of bases. Billions of these bases are all connected in very long pairs of strings. When cells divide, these paired strings need to be copied so a new set of DNA can be included in both cells. While copying these long strings of DNA, it isn't unusual for a cell to make a mistake. DNA can also be hurt or broken by things like exposure to too much sunlight or to certain chemicals. The good news is that our bodies have molecules that look for and help fix those mistakes. There are special proteins called enzymes that are always looking for mistakes or for damaged DNA. If they find it, they cut out that tiny part of the string of DNA. Then they replace it with a new, correct piece. Another important feature of DNA is that it has 2 strings that aren't the same, but that are a matching set. So if you have one string, you would know what the other string is supposed to look like. If only one string of DNA has a mistake, the other can be used to let the enzymes know what needs to be added. But if both strings have problems, both need to be replaced. A situation like this can be used to introduce new DNA. How did scientists figure this all out? For the last century or so, people have studied how DNA works. We learned that it passes on genetic information, and we know what it looks like. We also learned which genes are involved in certain diseases and which genes cause specific traits. There are even kits you can get to gather a sample of your own DNA to send in to be tested. You can see your risks of disease or learn about your ancestors. Once scientists knew the basics of how genes worked, the next step was to figure out how to change or modify specific genes. You may not know it, but humans have been modifying the genes of other organisms for thousands of years. But in the past, we've done this by picking which animals or plants to breed, by exposing eggs or seeds to radiation, or by adding genes to DNA at random. In the late 1900s, scientists discovered ways to carefully insert specific genes in exact locations in DNA. But this was very difficult and expensive to do until recently. A newer method called CRISPR has made gene editing much faster and cheaper. CRISPR has 2 main parts, a piece of RNA called guide RNA or gRNA, and an enzyme that cuts DNA. The gRNA can be made to find and connect to a specific place on a strand of DNA. Once the gRNA has lined up the CRISPR system on the DNA, the enzyme cuts the DNA. The enzyme used most often as a part of CRISPR is called Cas9. Once the DNA has been cut, the cell's own response of trying to fix it takes over. The cell will start to repair the cut. It uses either a remaining strand of DNA, a recent copy made from the DNA if it is still in the cell, or it will just rejoin cut ends of DNA. But if scientists introduce a strand of DNA that they've designed, it could be used as the instructions to repair the damage. This means a new gene that wasn't there before could be made and added to the DNA. In this way, CRISPR-Cas9 has been used to fix genes, remove genes, and add genes. It can also make genes stop working temporarily, which we call gene silencing, or stop working altogether, which we call a gene knockout. There are many ways the CRISPR system can be used. It could help avoid or cure disease, repair DNA damage, or even make existing genes work better. But taken further, those tools could also be used to make designer babies or to make microscopic weapons like drug-resistant bacteria. CRISPR technology is here to stay, and for good reason, but there is an ongoing debate about what should be off-limits for gene editing. In the comic Spider-Man, after Peter Parker's genes are modified, giving him superpowers, his uncle tells him something that rings true in real life now. With great power comes great responsibility.
We'll finish our tour of middle school science at Stanton Middle School. 8th grade teacher Atia Harris and her students have been creating models of energy flow and matter cycling through ecosystems. Here, see for yourself. Energy is an important theme in 8th grade science. Besides energy flow through ecosystems, students learn about energy transfers through waves and energy transfers in the atmosphere that affect our weather and climate. Engineering projects in 8th grade include designing structures that stand up to ocean waves, as well as structures that withstand destruction from severe weather.
I want you in groups of 3. So we got 1, 2, 3, 1, 2, 3. If you have read the directions for part 2, clap. That sounds a little bit better.
So Ms. Harris, can you tell us a little of what we're going to be seeing today?
Well, today we have been actually working on energy pyramids. So we've been talking about how energy transfers from one trophic level to the next. We've been using the rule of 10%. So we've been doing different formulas with math and things of that sort. But we use these big numbers, so they don't really get the opportunity to see what 10% actually looks like. So in today's lab, they're going to actually see how energy moves, and it's not a lot of energy that goes to the next level. So I think they really get an idea that 10% is not a lot, and that's why energy decreases as we move up.
So what
the
does number
energy
1 tell
pyramid.
us to do? Observe the experimental data in table 1. You see that? And it tells us the available energy and trophic levels of the cedar glade, okay? So that is an ecosystem that we're going to be looking at the data from that ecosystem, all right? So it tells us in trophic level number 1, how much energy are we starting off with? 200, good. And then in trophic level 2, primary consumers, how much energy are we starting off with? 19.6. Trophic level number 3, how much energy? Good. And trophic level 4, 0.19. So this is the rule of 10%. So we talked about the 10% rule, correct? So only about 10% of the energy at any given level is transferred to the next. Okay. So we're talking about energy pyramids. Remember how much energy is transferred from one trophic level to the next? 10%. So what we're going to do is we're going to actually do that, and we're going to actually see that 10% by doing this lab. With majority of kids, because I try to ask them at the beginning of the year, what do you guys like? What do you don't like? What can I do to make science fun for you? And everyone always say labs, 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 labs. So now we're going to actually see what that looks like. So if you have been designated as the material person, to get the material, at this time, I want you to come and get the flask. So the next thing I need you to get is the 3 clear cups. And once you get the 3 clear cups, we're going to label those in a moment, okay? So let's label our equipment so we know exactly what it is and what we're looking at as we're moving forward in the lab, all right? Good. I need you guys to put 2 drops. Remember, we're going to do 10%, and what does 10% look like coming off of this 100 milliliters? It gives us a formula. It says 10% of 100 milliliters equals how much?
With TCI, we're very focused on vocabulary and writing, so we try to incorporate the different labs in the hands-on and modeling and demonstrating to help engage them so that not only can they read it, now that they can actually see the science aspect to what they're doing and while they're reading and writing the science.